Good evening, all. It is my privilege on behalf of CAHO. I welcome Dr. Navin Kumar, the second speaker of today's topic, the quality control in molecular testing microbiology. Dr. Navin Kumar is a consultant and head of clinical microbiology and infection prevention at HCMCT Manipal Hospital in New Delhi. Dr. Navin Kumar, an experienced clinical consultant, a diverse background, having worked in both India and in the United Kingdom. His experience spans clinical microbiology, good clinical laboratory practices, epidemiology, infection control, and medical education. His field of expertise, you can say it's wide: bacteriology, serology, virology, immunology, and quality assurance in microbiology laboratory. And also molecular diagnosis of infectious diseases, good clinical laboratory practices, hospital infection control, infection management in critical care units, and organ transplant patients. Of course, antimicrobial stewardship, nowadays we are all talking about it. It's a very important thing. And to his credit, Dr. Navin Kumar has a lot of awards and achievements. To his credit, he is an empaneled assessor of National Accreditation Board for Testing and Calibrating Laboratories, that is NABL, in India for microbiology and molecular biology laboratories, and is a certificate of attainment of antimicrobial stewardship from the University of Dundee, UK, and empaneled assessor entry level for NABH and Quality Council of India. So it is our privilege to welcome Dr. Navin Kumar on behalf of the CAHO, and it's a very important topic. Let us know more about quality control in molecular testing, microbiology. Over to you, Dr. Navin Kumar. Thank you.
Thank you, sir, for such a kind introductions. So good evening, everyone. My talk will be on essentials of setting up and running a molecular laboratory. And we know that molecular testing is rapidly expanding field in infectious disease diagnostics, offering advanced techniques, methodology, and applications. During the COVID-19 pandemic, the molecular techniques became the primary mode for detecting the virus. This brought new responsibility for clinical microbiology laboratories. Today, clinical microbiologists are expected to understand various molecular techniques and take a leading role in managing molecular testing laboratories. So when we talk about molecular infectious disease laboratories, the following are the key requirements and important factors to consider. And first is the good laboratory design, adequate and appropriate equipment, good assay design, workflow considerations, and manpower training. So when we're talking about laboratory design, spatial separation of work area becomes very important. And we all know that molecular infectious disease laboratory should ideally be separated into 3 areas. Area 1 should be known as reagent preparations and reagent storage area. Area 2 is for the specimen preparations or nucleic acid extractions. And area 3 is for amplification process. So there should be these 3 areas should be physically separated and preferably at a substantial distance from each other. So this is the pictorial representation of the the 3 areas. So area 1, which is the reagent preparations or master mix preparation area, and should have positive air pressure compared with other rooms. This is the cleanest area of the molecular lab. Specimens or prepared nucleic acid should not be brought into this space. The second one, which is the nucleic acid isolation or a nucleic acid extraction area, and SS setup is also happening in the second area. So this room is under negative pressure to retain all aerosols within the area. So it means that in comparison to corridor or other rooms, the airflow should be inward into the negative, into the second space or second area. The third one, which is the amplification room, which we call it, and it is for the amplification and detection analysis happens. It should be again in the negative pressure compared to the the corridor, and it's basically it's the negative pressure is required to minimize amplicon contamination into the clean space. If space is limited, that's what we see in in a smaller lab, and many of the hospitals, when the hospital is already functioning lab and there is a space constraint all the time. So in those setup, in where the smaller lab and there is space is limitation, the sample preparation and reagent preparation can be performed in same room, and it should be considered as clean space, but should be separated as much as possible. Because in molecular infectious disease diagnostic laboratory, it is essential to ensure proper attention to personal safety and workflow to minimize the risk of contamination by amplicon and cross contamination of patient specimens. Now, next is equipment. So, molecular laboratory is equipped with basic instrumentation, but also with unique equipment for nucleic acid, amplifications and downstream detections. And these equipments are thermal cyclers, real-time PCR units, centrifuges, vortex, micropipettes, capillary electrophoresis unit, micro air instruments. Depends upon the the volume of, depends upon the type of test and the volume of test. There should be separate sets of equipment and supplies for each area. And also, the wide variety of instrument placed in the molecular lab ideally should be placed in accordance with the physical layout of the lab, having adequate space and ventilations. The other important point need to consider is the positive displacement pipettes with filtered tips, and it should be used for all pre-amplification procedures. The filter tips minimize the potential for carryover from a specimen to a specimen. And the last point is also very important, which is molecular laboratories should use manual defrost freezers, because self-defrosting systems causes temperature fluctuations and solvent evaporations with each freeze-thaw cycle and are not appropriate for storage of reagents. So preventive maintenance schedule should include manual defrosting of these fridges. Now continuing with the equipment, Class 2 biosafety cabinet for sample preparations and nucleic acid extraction is critical for contamination control, personal safety, and product integrity in a molecular lab. And then laminar flow cabinet can be used for master mix preparations and target loading. Now, when we're talking about nucleic acid extractions, and now most of the lab has got automated extraction system, the instrument should be selected based on their design to minimize carryover between samples and performance in purifying nucleic acid. Carryover studies in the format of checkerboard of high positive and negative should be performed as part of any extractor which we are going to use in the lab. It is preferred that automated platform use disposable reservoir with easy to remove waste and clean waste container. Now, other issue arises because in most of the lab, on the molecular lab now, is there are a lot of sample to result instrument are in use. For example, if you see BioFire, QIAstat, and also Gene Experts, and most of the labs are performing various tests on these sample to result instrument. They have really made a good contribution in terms of, because it reduces risk of contaminations. And also the question arises where this equipment should be kept in the lab. If we have enough space, then the separate area can be created for keeping these instruments, but sample to result instrument can be placed in sub-contained area within the amplification room. Now, next is the assay design. I think most of the labs now are using, the commercial kits, which has already been, like master prepared master mix are already there, and very minimal addition are done in the lab. But the homegrown tests are also being used. So whatever we need to have some understanding about assays when we are selecting any kits for our use. So target selection, and for the target selection should have, the assay should have highly conserved and pathogen specific gene region to ensure a sensitivity and a specificity. Conserved regions are part of pathogen's genome, and it do not change between different strains. So choosing conserved region ensure that assay can detect all variants. For example, Mycobacterium tuberculosis, the insertion sequence 6110 is often targeted because it is present in multiple copies in most Mycobacterium tuberculosis strains, so increasing the sensitivity of detection. Gene targets are also selected based specific to pathogen to avoid false positive. And good example is the recent SARS-CoV-2 PCR, where we have commonly targeted genes include E genes, N genes, nucleocapsid, RNA-dependent RNA polymerase, and spike protein. So different assay has got different combination of the different genes of SARS-CoV-2. Now, primer and probe design, when we are, we also need to consider that primer and probes are short sequences of nucleotides that bind specifically to the target DNA and RNA. So correct design of primer and probes are essential for test accuracy. Primer must bind only to target sequence and not to human genome or unrelated microbes. The melting temperature should be similar between primer and probes to ensure effective amplifications. And also, the primer probes should, such as should avoid secondary structures such as hairpins or dimers, which reduces the efficiency of the assay. And the GC content, which is it's ideally it should be 40 to 60% for a stable binding for a good assay. When we are introducing new molecular assays, some design consideration can help in minimizing the contamination and detection of contaminations. So, frequency and location of negative control depends upon the assay run size, positivity rate, and nucleic acid concentration of positive specimens. So single negative control is sufficient for assays with a small number of specimens per run, but in a high throughput lab and with a high positivity rate, for example, if negative control every 10 samples for a run size of more than 20 specimens, or if a assay which has got high positivity rate of more than 20%. So these are some of the things which we need to consider when we are designing or introducing an assay in our lab. Now, next is the workflow consideration that is very important. So, testing in molecular laboratory should always follow unidirectional workflow. It means that nothing should come back from the amplification room to sample preparation or reagent preparation room. So unidirectional flow from reagent, from sample preparations to extraction to assay setup to amplification room. That's the flow should be followed. Then the another critical component in molecular laboratory is the proper cleaning of work areas. The benchtop equipment and furniture should be cleaned with 10% sodium hypochlorite followed by 70% alcohol. The sodium hypochlorite solution should be prepared fresh daily. Another, we need to remember the sodium hypochlorite is corrosive to plastics and metals, so we need to be careful about when we are applying on any equipment, and just follow the manufacturer instructions also that how the cleaning should be done for those equipments. There are commercial DNA RNA destroying cleaning solutions are available, but they are expensive. The other important point when we are, in terms of cleaning and disinfection, disinfection protocol in the lab, that same cleaning process should be followed after work is completed. So at least two-time cleaning is required in a molecular lab, and not just at the beginning of the lab and leave it at the end. So that after we finish our work, the cleaning process should be followed and should be done as we have done at the beginning of the work. UV lights may be used for secondary decontamination of, uh, for PCR workstations, and UV light works by photocross-linking pyrimidine and thymidine in the template DNA, creating a non-amplifiable product. The other thing which we need to, when we work in the lab, and also it is for the good laboratory practices, the racks that are reused should be cleaned by soaking in 10% bleach solutions or 10% hypochlorite, you can say. Any racks or equipment removed for amplification area first be decontaminated using bleach and alcohol, because these are the points I have added on, because these are always been overlooked. Cleaning of door knobs and handle of freezers is also should be part of the cleaning and cleaning protocol of the lab. Gloves should be changed when contaminated or when moving between the different areas. Hairnet and safety glasses should be used appropriately. Dedicated lab coat or gown, disposable preferred if lab can afford, for each PCR room. So that is also very important. Wearing one lab coat, we should not be going in from one area to another area. So it is also preferred because that all reagents be prepared in single-use aliquots. Sample preparations or nucleic acid extractions steps carry high risk of contamination. So some specimens should be pipetted using filter tips that should be changed between specimens. If specimens are being pipetted from deeper tubes, then extra long tips are recommended. So these things, small things, we need to be cautious about as part of the good laboratory practices in molecular lab. Specimens should be opened one at a time to avoid cross contamination. Closed amplification system minimizes the risk of amplicon contaminations. If there are post PCR processes happening in the lab, if the lab is doing NGS or Sanger sequencing or gel loading, then that are at a high risk of contamination, and additional workspace need to be provided for opening of amplification tube. This area, be thoroughly decontaminated. Rack should be cleaned before leaving that room. Now, another important part, and it is very, always been, again, underlooked or, overlooked or not been followed, is monitoring of laboratory for contamination. Because contamination is the biggest, what to say, I can say biggest r- risk in a high throughput lab, and it goes unnoticed unless it happens. We cannot, see the contamination by naked eye. So once it happens, it really gives a bad name to the lab, and a lot of the retesting. And so that's why the contaminant monitoring of laboratory for contamination is very important. Contamination is introduction of unwanted nucleic acid into the specimen. Repeated amplification of same target sequence leads to accumulation of amplification products in the laboratory environment, and buildup of aerosolized amplification targets will contaminate laboratory reagents, equipment, and ventilation system. So swipe or surveillance testing, we can call it, is a common method of monitoring a laboratory for contaminations. So for swipe testing, Dacron swab should be used to swipe all three areas of the laboratory, and frequency of swipe testing may vary by the season, analyte, and the laboratory. Laboratory with a high volume, high positivity rate of tests should perform swipe testing weekly. Laboratory with lower volume of testing or tests with lower positivity rates may perform swipe testing monthly. But every analyte that is being amplified in the lab should be tested at least 3 times a year. So we need to define our swipe testing depends upon our lab test menu. Result of swipe testing should be documented and reviewed. And if there is a contamination detected by the swipe test, then area may be swiped in smaller sections to identify the source of contaminations. Now, this point, the manpower training and training is very important in molecular lab. Molecular testing is new field in diagnostics with rapidly expanding information. So the molecular laboratory personnel need to have overall depth of knowledge and understanding. The quality management system of the lab should establish an education program to ensure that laboratory personnel understand all aspects of testing. They must be trained not only in technique but also in contamination control, result interpretation, and equipment handling. Molecular infectious disease assay results may require explaining the significance of results and its application to the physicians who are sometimes unfamiliar with the methodology. So, laboratory clinical staffs need to understand the technical component of the technical component information like clinical validity, clinical utility, and ethical issues about the assay. And a structured competency and continuing education program should be established in the lab, utilizing internal and external resources. This is an example of competency assessment checklist, which I have just shared here. And in a competency in molecular testing should be assessed in the following area. It should include, all aspects of the molecular testing like cleaning, disinfections of work area, pipetting of samples, manual extractions of samples. If lab is doing preparation of master mix, unidirectional flow in the molecular is followed or not, assay setup, and handles of other amplification equipments, and also how the results are read by the laboratory personnel and if the personnel can interpret the results appropriately or not. So basically, the competency assessment for molecular testing should be separate from your routine laboratory competency assessment. And the tool is the similar which we use for the other competency assessment, and that's how the level of competence is documented. So these are the specific molecular educational informations which can be obtained from following resources, so that can you can gain ongoing CMEs, ongoing webinars on these resources as well, and try to remain update with the development in the molecular testing. I'm just going to escape the slides because a lot been talked about the Dr. Shah. So now coming to the summary of my presentations. Good laboratory design, careful attention to the workflow, and establishment of effective surveillance mechanism ensure a high level of quality in molecular infectious disease test results. Thank you very much.
Thank you, Dr. Navin Kumar, for the lucid presentation. Very well taken. The quality testing in molecular testing, especially in microbiology.